Hello everyone, my name is Berrrebi from Vanier College, a technical institution in Montreal, Canada for pre-university education. I will be presenting the exploratory analysis I collaborated on with Dr. Ferreira from the University Rio Grande do Norte in Brazil on the behavioral stability of Capuchin monkeys. The broad objective of this analysis was to investigate which axes were most stable in the personality of individuals. A minor objective was to determine which sex presents with a more stable behavioral profile overall. We used Reale's definition of personality, being the stable inter-individual differences that persist over time and various situations, as the foundation upon which we laid our work, as it focuses on the differences in mean behavior values among individuals. However, it is known that individuals' behavior varies along days as well. We are interested in comparing the differences among individuals with those within an individual, as it will provide a more comprehensive perspective on the stability of behavioral profiles. Capuchin monkeys are interesting models in comparative psychology due to the increased sizes of their neocortexes, as well as their ability to manipulate tools. They are good models to test the evolution of personality, as they have characteristics that intersect with hominids, the group that humans belong to. 33 wild-born, rescued adult capuchin monkeys were studied for one academic semester, during which each individual was observed daily over 5-minute intervals. My co-authors at Dr. Ferreira's Rescue Center noted how many seconds were spent engaged in different activities, filtered into the following 6 behavior macro categories, which represent 5 possible personality axes: feeding for food-oriented, environmental manipulation for exploration avoidance, positive sociability and solitary play falling under sociability, vigilance for alertness, and locomotion for activity. The data collection process was behavioral coding. These groups had stable composition and observations were always conducted in the afternoon, so to maintain consistency throughout the observations. On to data analysis. We used the mean values of percentage of each individual's behavior to test the AIV among individual variation and the standard deviations from these mean values to test the intra-individual variation IIV. For statistical analyses, we compared the standard values, the z-scores, for AIV and IIV respectively. Normality tests were conducted to verify the data's adequacy, and student t-tests were used to compare males and females respectively as well. Full disclosure, the data analysis was conducted manually via the statistical package PAST. Right from the get-go, I can tell you that males and females did not differ in their behavioral profiles, which can be very much seen in this figure here, except for environmental manipulation and in vigilance. Environmental manipulation, males manipulated more than females, which was expected, seeing as in the wild males crack open nuts with tools, and for vigilance, females were noticeably more alert than males, which once more we expected given the previous data that was available. Here we see that for AIV and IIV, we hypothesized, firstly, that given the groups of normal raising conditions, there would be a lot more AIV than IIV. However, we see the opposite, that the variation among individuals is slight. There are axes that present with great IIV, with either equal or larger than that of AIV. Those axes are feeding, vigilance, and locomotion. When comparing females and males, there were no noticeable differences in AIV and IIV values, indicating variation among individuals is similar to the variation within individuals for both sexes. Thus, we found no statistically significant differences for both sexes for most axes. When we compared the standard scores, we found that AIV z-score values were small. Here we see that feeding, locomotion, and vigilance exhibit the largest amount of among individual variation, whereas social positive, solitary play, and environmental manipulation exhibited the smallest amount of among individual variation. Contrarily, IIV z-score values were large, which is made evident in the figure as all axes but one, locomotion, had individuals with variation surpassing 2 standard deviations. We see for feeding, for social positive, for solitary play, environmental manipulation, as well as vigilance. When comparing the IIV and AIV z-score graphs side by side, we can see a stark contrast in most axes among the two. The slight AIV and large IIV provides evidence for a great amount of behavioral plasticity within this group of monkeys. The small AIV values could be explained by the captive environment, as it does not foster much variance. The large IIV differences, however, may be due to the animal's abnormal raising conditions, seeing as they are rescued. The small variation among individuals, in combination with the large intra-individual variation, expands discussions on the stability of personality, thus pointing to significant behavioral plasticity for this group of monkeys. As such, we recommend that more research be conducted in the wild and with the implementation of differing data collection methods. Finally, I'd like to sincerely thank everyone who helped me throughout this entire process, most importantly Dr. Ferreira, who has been my mentor since the beginning. I'd also like to thank Professor Ruffalo from Banya College, in addition to the co-authors that conducted all the observations on site and assisted me throughout the process. Thank you very much. I'd now like to open the floor to any questions that you might have. I will try my best to answer them. Thank you very much. I'd now like to open the floor to anyone who might have any questions. I will try to answer them to the best of my ability.